Welcome to the University of York. This is the Department of Chemistry and my name is Richard Gammons. And today I'm going to talk to you about how we go from plant biomass to biofuels. The first part is plant biomass. Here in front of me we have a selection of biomass materials we can use. This ranges from common food sources like wheat to grass materials like the miscanthus. Now we want to try and avoid the food-based materials and use materials that aren't for food when it comes down to our research here. Once we've got these biomass materials, they will obviously look a bit like this. This is an example of some maize stover from the field. Before we can use this, we have to grind it down in bore mills so it resembles a fine powder material that we can use in our work. The next part of the process is the pre-treatment part, and this part is to open up the plant cell wall to get access to the cellulose, hemicellulose and lignin therein. One way of doing this is using concentrators of uric acid, and this breaks apart those hydrogen bonds and allows access to the polymers. Another way is using steam explosion, and this involves using high pressured water and high temperature water to break apart the polymers. In the lab, we can do this using a hydrothermal bomb. By heating this up and sealing this steel lid closed, we can create high pressures. So here at the University of York, we're trying new base solvents to do this pre-treatment. And in front of me, I have an acid and a base, which creates a new compound. If we open the separation flask, we can see as the acid goes in, the reaction taking place is exothermic, releasing HCl gas and water. Once we've pre-treated our biomass, we're left with a sample that we can now use for enzyme hydrolysis. We can take an enzyme mixture, in this case cellulase enzymes to digest cellulose, and put it either in well plates, as we have here, or we can have lots of samples, do lots of replicates to make sure the data we get is accurate, or we can do individual studies by placing them in these vials as such. The enzyme mixture and the biomass is incubated at 50 degrees for 8 hours to hopefully produce large quantities of sugars, in this case glucose. Now we have our sugars from the polymers in biomass, we need to get to bioethanol, and this is done by fermentation. One molecule of glucose is fermented into 2 molecules of ethanol and 2 molecules of CO2. This is done by using yeast. First of all, we have to grow the yeast on agar plates to produce yeast colonies. From these colonies, we can add these to the fermentation vessel and add our biomass. Then, bioethanol is produced and the CO2 is dissolved in the water above. This microfilm, so the conditions are sterile, so we don't kill the microorganism yeast in the process. Now we have a mixture of biomass, some sugars, ethanol, and the medium that the yeast is in, as well as some buffer. To get our ethanol from the solution, we have to do a distillation. We can heat on the one side, cool down by our condenser to produce the clear liquid, which is ethanol. After that's complete, there you have your bioethanol from your plant biomass.